Hello and welcome to this Revision Monkey video on what's going to be in the Separate Biology Paper 2 exam in 2023. This is for pupils studying the AQA specification, and this is for the separate, or your school might call it triple, scientists who study the longer papers, which are 1 hour and 45 minutes long. Let's have a look at what to expect in Biology Paper 2. So again, we'll very quickly recap compare, explain and evaluate questions. We'll look at a little bit on graphs, a little bit on tables, because you need to expect lots of tables and graphs in your biology paper. We'll have a look at some of the calculations to expect. About 10% of the paper will be calculations for biology paper 2, and also 15% of the exam will be about required practicals, so we'll have a look over those ones as well. So as a quick reminder, for evaluate questions, they won't tell you this, but you must include advantages, disadvantages, and a justified conclusion in your answer. They will just have the word evaluate in the question, but it means give advantages, disadvantages, and a justified conclusion. Compare questions, again, they won't tell you, but when you answer compare questions, you need to write similarities as well as differences in your answer. And explain questions, you must say why something is happening, so include as much scientific detail as possible. So I do go into a bit more detail in these in the first biology video, so feel free to look back there if you want to look at these evaluate, compare and explain in more detail. So in biology paper 1 we did a little bit of work on graphs. Just to add to that, you need to read the graphs really carefully. So if the question asks what is the rate of photosynthesis when the light intensity is 400 lumens, they'd expect you to show you're working on the graph by drawing a line up from 400. But my point is, if when you draw the line up, you meet the line on the graph and it is not exactly on the corner of a square, that is okay. Just continue by drawing a line across. And you can see this one, it doesn't come off at 30, it doesn't come off at 31, so we need to be really accurate that our answer is going to be 30.5. Don't force it to meet one of the lines on the graph. If you find that it comes in the middle of a box, that's absolutely fine. Just read it accurately rather than forcing it up here to the next line. Also, I just want to look at describing graphs. So this one says describe the relationship between light intensity and the rate of photosynthesis. So you're looking at 3 different shapes on the graph. First bit is the steep bit at the start. So you describe that by saying between 0 and 300 lumens as light intensity increases, the rate of photosynthesis increases rapidly. So there's the key word there, underlined, rapidly. And then you have to look at the next part of the graph, and between 300 and 560 lumens, as light intensity increases, the rate of photosynthesis increases gradually. So this is a more shallow line, so we call it a gradual increase. And then finally, looking at the last part of the line, after 560 lumens, as light intensity increases, the rate of photosynthesis stays constant. So a really, really common question for biology paper 2, but it could come up in chemistry or physics as well. There's more information on graphs in biology paper 1, but I'll assume you've had a look through that already. Again, I did a lot of examples of tables in biology paper 1 video, but I just wanted to add that they might give you a blank table to fill in. So they give you all the information in the question and ask you to complete a blank table. So this question says a student carried out a transect. They placed a quadrat every 50 centimetres from a tree, and they counted the number of daisies in each quadrat, and their results are below, and they'll give you a list of results. And then it says complete the table to show the student's results. One result has been completed for you. Sometimes they'll complete the first result to give you a clue to put your headings in the table, and sometimes they don't, so look out for that. If they don't, you must make sure you've got the headings in. So you'd have to complete the table by writing in appropriate headings. So it said they placed the quadrat different distances from the trunk, so let's call this distance from the trunk in centimetres, And they counted the number of daisies, so you put that in the right-hand column, and you're going up in 50s as the question tells you, and in this column you're just writing in the order that they've given you the results as they have collected them. But again, there's more on tables in paper 1 if you didn't get to watch that video. Have a look back and watch those first few minutes. 10% of biology paper 2 is going to be calculations. So this is a calculation which is linked to a required practical, so I just wanted to go over this one. It's about estimating population size. So the question says, estimate the population size of snails in the 30 metres by 25 metre woodland. That's represented over here. And the student used a 0.25 metres squared quadrat, and there were a mean of 3 snails per quadrat. So just bear in mind, the quadrat, if it's got metres squared, that's already the area. You don't have to do anything with that, it's already the area. If they give you the quadrat as a diagram like this, you might have to work out the area by multiplying 0.5 by 0.5, as would be this example. So if it's got metres squared, the area has already been calculated for you. If they give you a diagram to show you the quadrat, you would need to calculate the area first by multiplying the length of both sides. So remind of the equation that we need to use is population size is equal to field area divided by quadrat area multiplied by the mean number of organisms per quadrat. Because this first bit will tell you the number of quadrats that would fit in a field. So the field area divided by the quadrat area would tell you how many of these quadrats would fit side by side in the field. And then if there was a mean number of 3 snails per quadrat, multiply that by 3, and that will give you an estimation of the population size. So if we have a look and calculate the field area, to give you 750 metres squared, we've got the quadrat area already, the number of organisms are 3, so putting our numbers in, field area 750 divided by 0.25 multiplied by 3, showing our workings out and ending up with our population size of 9,000 in this example. So look out for that one if that one comes up in the paper 2 exam. Also, uncertainty can come up on any paper. Uncertainty is calculated by the range divided by 2. For example, this is a set of results with 2 people, A and B, and it measured the distance the ruler fell through their hand. So they've taken 3 repeats and calculated the mean. You'll notice the mean is the same for both people, but the uncertainty will be different. So uncertainty in the person A's case would be 7 minus 5 divided by 2, because the biggest number is 7, smallest number is 5. That gives us our range, and then divided by 2. So our uncertainty would be plus or minus 1. And then the uncertainty for the second set of data would be plus or minus 4, because the biggest number is 10, smallest number is 2, and that would give us our range, 10 minus 2 divided by 2. So yes, they both have the same mean, but because the person A's numbers were closer together, they have a smaller uncertainty. Because person B's had a bigger range in their results, they have a much larger uncertainty around their mean. So the 2 calculations to be aware of, uncertainty can come up in any paper, and population size calculation will just be relevant for biology paper 2. So the required practicals for separate science are organism distribution, so quadrats and transects, reaction time required practical, the decay required practical, and the plant growth required practical.
Reaction time required practical. As reaction time is so quick, it's very difficult to measure with a stopwatch. So instead, we're going to measure reaction time using the ruler drop test. For example, we could ask the question, how does caffeine affect reaction time? Now, this is just one example of a factor that we could investigate. You could well in the exam be asked about something like distractions or tiredness or something like that. So this here could change and become a different factor. So let's go through the method. First of all, you'd work in pairs for this practical. Person A will be the person who is dropping the ruler, and person B will be the person who is catching the ruler. Person B should sit down and rest their dominant hand on the table with their thumb and fingers hanging over the table. Person A should hold the ruler vertically with the base of the ruler in between person B's thumb and forefinger. This is an important control variable because you need to make sure that you drop the ruler from the same height every single time. Person A will drop the ruler without indicating to person B when this will be, and person B should catch the ruler as quickly as possible. If the ruler falls through just a short distance, this would indicate a fast reaction time, and if the ruler fell through a longer distance, that would indicate a slow reaction time. We can then use a conversion table to convert the distance the ruler fell through the hands into a reaction time. So this will turn the centimetres from the ruler into a time in seconds. We would then repeat this 3 times and calculate a mean reaction time. So you've got the reaction time for person B. We're then going to change this factor here, the amount of caffeine they've drunk, and then we're going to repeat the experiment. So the next step, person B should then drink a set volume. It doesn't matter how much you put, but I've just suggested here 200 mils of caffeinated Coke. They should wait 5 minutes. Again, you should just suggest a sensible amount of time here, and then repeat the ruler drop test 3 times and calculate a mean reaction time. Repeat the investigation using 10 other people, so you could first swap around so that person A and B swap around, and then find other people to repeat the investigation.
So if we look at some variables for the investigation, the independent variable would be the volume of the caffeinated drink that was consumed. So in this case, for example, 200 ml or 0 ml were the choices. The dependent variable would be the reaction time, because that's what we're collecting data on. And the control variables, things such as using the same hand when catching the ruler, and also making sure the ruler is dropped from the same height.
Organism distribution required practical. There are 2 different ways that we are going to look at to sample organisms. The first one is random sampling, and the second one is transects. So random sampling might be used to estimate a particular population size. So for example, you might get a question a little bit like this that says, describe a method to estimate the population size of daisies growing in a 200 metre by 100 metre school field. These numbers here will come up a little bit later on in our calculation. So we can't count every single daisy growing in the school field. It would just take too long. So we need to have a method of sampling that is completely random, and if they talk about why in the exam, it is to avoid bias. So as we said, we can't count the number of daisies in the whole 200 metre by 100 metre field, so instead we measure out an area of the field using a tape measure. So here I've got a 5 metre by 5 metre area, for example. We then divide this grid up into squares using string and number those squares 1 to 100. So our grid might look something like this. Then the most important thing with random sampling is we use a grid called a quadrat. Now the one I'm going to use in this example is a 0.5 metre by 0.5 metre quadrat. In the exam it may well be a different size, but that's absolutely fine. And we need to choose where to place our quadrat within this grid. So again, it would take too long to do all 100 squares and count the number of daisies in each square. So what we use is a random number generator to select a square, and you can get those on calculators or phones, and it will pick a number at random for you. So we place a quadrat on that selected square and count the number of daisies present, and then we repeat the steps for perhaps 9 more random squares. So we might do 10 in total. So let's say our random number generator picked us out the number 9. What we do is we take our quadrat and we place that on square 9 and we count the number of daisies present. We would then repeat that for another square. For example, if our random number generator there picked us up 47, we'd place our quadrat on 47, count the number of daisies present, and again, for example, 72, again counting the number of daisies present. Now we'd repeat that for around 10 squares. So we've got different quadrats here, and we've counted the number of daisies present in the squares. And we're going to then calculate a mean number of daisies per quadrat. And because we're going to use this number again in a minute in a calculation, I'm just going to leave it as a decimal rather than rounding it as I would do normally with a mean. So we've got a mean number of daisies per quadrat, and now we need to use this to estimate the number of daisies in the whole school field. So we need to know our area of our quadrat, so we do 0.5 times 0.5, and that's 0.25 metres squared. We need to know the area of the school field, so given in the question it was 200 metres by 100 metres, so 20,000 metres squared. The next thing that you need to work out is the number of quadrats that would fit in the field. So we've got the area of our field divided by the area of one quadrat, and that gives us 80,000 quadrats. So if 80,000 quadrats fitted in our field, and the number of daisies in each quadrat was 0.8 as an average, then the estimated number of daisies in the whole field would be our number per quadrat multiplied by the number of quadrats that would fit in the field. So 0.8 times by 80,000 would give us a total estimate of 64,000 daisies. A couple of key points then. Increasing the number of squares sampled will increase the reliability of the population size estimate. They may use the word reliability or they may use the word accuracy, but the more quadrats that you put down on that field, the more reliable your estimate is going to be. Now, for some organisms, calculating percentage cover might be more appropriate than counting the number of organisms. For example, you may well have seen something like lichen growing on trees or walls. If you wanted to sample lichen, you might use a really small quadrat, so just perhaps a 10 by 10 centimetre grid. In the previous example with daisies, we would look at one random square which we'd selected by our random number generator, and we calculate the number of organisms inside. However, with lichen, we can't do this because you can't see individual organisms. So instead, you might need to count the number of squares covered by the lichen. Now, all of the whole squares you can just count up like so, and then we will look at any partial squares that we've got, like perhaps this one and this one, where they could fit together roughly to make a complete square. And we will continue to do this, estimating the total number of squares that are covered by the lichen. So when we've got this, we use the equation the number of squares covered divided by the total number of squares times 100, and this will give us our percentage cover. So if I've estimated that 30 squares in total are covered by the lichen, there are 100 squares on the grid, so 30 divided by 100 times 100 will give us an answer of 30% coverage. Another way we could sample the distribution of organisms is using a transect. So for this we might be asked questions such as, how does light intensity affect organism distribution? Or, how does the distribution of organisms change as you get further from the shore? So let's take this example in terms of seeing how light intensity affects organism distribution. So under the tree, the organisms will be subject to more shade than further away from the tree. So we can sample the organisms at regular intervals and see how the light intensity affects their distribution. So what we do is we'd lay a transect line, this could be a tape measure or some string, along the ground starting at the base of the tree. We would place a quadrat at the 0 metres mark and count the number of organisms, so for example daisies, or it could be something else that you're looking at, present in the quadrat. We would use a light meter to take a recording of the light intensity and record this in a table. We then repeat steps 2 and 3 at regular intervals along the transect line, for example every 2 metres. So we place our quadrat and our light meter here, 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 each time counting the number of organisms, for example daisies or whatever we were looking at, and calculating the light intensity. Now I want you to imagine that we're sampling in a woodland, and along the transect away from the tree there's lots of long plants and long grasses. Now in this case we simply can't place a quadrat on top of the long grass, okay, because the grass is just far too long. So you might see in the exam them talking about counting the plants that are just touching the transect line. So if you've used string and pulled that along the long grasses, you might be simply counting the different species that are touching the transect line. Another thing to look out for, don't be put off if they talk about wanting to measure something vertically. So for example they could look at how height affects leaf size or how height affects lichen coverage. In this case, think about doing the practical in exactly the same way. So you may use a very small quadrat and measure percentage cover of something as you go up, or you might simply, if you're measuring leaf size for example, see any leaves that were touching the line and measure their size as you go up and see how that changes as you go up the transect. So don't be put off if they're talking about one vertically going up a tree or up a wall or something like that. It's exactly the same process as the horizontal transect that we looked at before. Plant growth require practical, and this is for separate scientists only. When you're looking at plant growth in a lab, you're going to want to use a seed such as mustard seeds or cress seeds that are going to grow very quickly. There are a number of different factors that you could investigate to see the effect that they have on plant growth. I'm going to go through a few of those, but you have to be resilient in the exam because they could talk about a number of different factors. The first one we'll look at is this question, which says, how does light intensity affect plant growth? So for this practical, we would set up a petri dish, and inside that petri dish you could have a small layer of soil, or you could simply just have some damp cotton wool. Seeds will grow very easily, so cotton wool would be absolutely fine. And you would put a certain number of seeds on there, making sure you keep that constant. So I've put 5 seeds on the dish, and if we're looking at light intensity, we could change the distance between the lamp and the petri dish, for example. So our independent variable would be the distance between the light and the petri dish, and this would therefore change the light intensity reaching the plant. Our dependent variable would be the height of the plant, and control variables would include the same number of seeds per petri dish, the power of the lamp, the type of seed that we're using, the volume of water that we add daily, and the temperature. And depending on the practical, we might want to measure the rate of growth, and we may want to measure the height of the plant every day, or we may just want to leave it for a set number of days and measure the height of the plants afterwards. So to measure the height of the plant, you'd put your ruler so it's touching the cotton wool or the soil or whatever you're using, and hold the plant gently. So you can see at the moment the one that the ruler's next to is slightly bent, so you'd hold it gently up against the ruler to get an accurate height. So you'd measure the height of each of the seedlings in each of the discs, and you'd have an average height that the plants have grown under each light intensity, which you would then record in a table. A different question that you might want to ask might be, how does the direction of light affect plant growth? So this time keeping the distance between the lamp and the petri dish the same, but changing the direction. So perhaps having the light coming in from the left or the right, or from directly above, for example. So in this case, our independent variable would be our direction of the light source. Our dependent variable might be recording the direction of the plant growth. Our control variables, important things as before, like the same number of seeds per petri dish, the power of the lamp, the type of seed, the volume of water added, temperature, and in this case the addition of an extra one, which is the distance between the lamp and the seeds. And then because of phototropism and the response to light, we would have the plant growing and changing direction towards the light source. And in the exam you may well be asked to draw drawings of the seeds to show how you might predict them to grow or as a result of a particular experiment, and in which case you would use clear pencil drawings. Again, linking into tropisms, this case geotropism, we might want to look at how the angle of the Petri dish affects plant growth. And in this case we might want to set up the Petri dishes at different angles and then record plant growth. In this case our independent variable would be our angle of our Petri dish, so we might want to do 180 degrees, 90 degrees, 270 degrees, etc. Our direction of our plant growth would be what we are going to record down after our experiment, and our control variables would be, again, similar things to before, and make sure we're keeping the direction of the light source the same, and also light intensity or background lighting we'd want to keep the same in this one as well. As a result of this experiment, then we may expect results a little bit like this, whereby the shoots are responding to gravity and growing upwards in all cases. Now you may well in the exam also see experiments where you can observe the direction of root growth as well, so look out for those ones too. Decay required practical for separate scientists only. In this investigation we're going to look at setting up an experiment to investigate how temperature affects the rate of decay. Now when we're looking at calculating rate, we're going to use an equation right at the end that says rate equals 1000 divided by time, because we're just looking at a colour change and this is the equation that we need to use. So for this experiment we're going to set up a few things first of all. We're going to set up 3 water baths, and I've just suggested these temperatures. It doesn't matter which ones that you suggest. So we've got one of 20, 40 and 60 degrees. If you wanted to have a cooler temperature as well, you may well set up some water in a fridge or some water with some ice in it to have a look at colder temperatures as well. But I'm going to look at one where we set up 3 water baths. And then for each water bath we need to set up 2 test tubes, or you can use boiling tubes, either is fine. One test tube containing 5 centimetres cubed of lipase, okay, so that will go in here. And the other test tube we're going to put in 5 centimetres cubed of milk, 5 drops of phenolphthalein, which is our indicator, and 7 centimetres of sodium carbonate solution. All of these measurements here we are going to measure out using a pipette, okay, because that can draw up a fixed volume and place it in our tubes for us. Now we're going to then place these into the different water baths. Now the sodium carbonate solution is something that makes the solution first of all an alkali. So with phenolphthalein it will go pink in alkali conditions. That was the closest colour I could get to pink, so we go pink in alkali conditions. And what will happen in this experiment is the lipase is an enzyme that will break down the fats in milk. And when fats are broken down, they are broken down by the lipase into fatty acids and glycerol. And it is this product here, the acids, that causes a colour change. So in the first instance, we have an alkali conditions created by the sodium carbonate, and then later on we're going to add the lipase, and that will break down the fats and produce fatty acids. So this solution here should go from pink to colourless, because that is the change that we have when we're using phenolphthalein. Now in the exam they may give you another indicator, or you may be used to using another indicator, and that's absolutely fine to do as well. So we've placed our 2 test tubes in our water bath here, and what we need to do then is put a thermometer into each test tube and wait for the temperature of each test tube to meet that of the water bath. So we just need to allow it to stabilise and get to the correct temperature. We are then going to remove 1 centimetres cubed of lipase from our lipase test tube using a pipette, and we're going to squirt it into the other test tube containing the milk, the sodium carbonate and the phenolphthalein indicator. Then, using a stopwatch, we're going to record the time it takes for the mixture to go colourless. And then we are going to repeat this for the other temperatures that we said before. We'd chosen 40 degrees and 60 degrees. And as always is good practice, repeat the experiment 2 more times for each temperature and calculate a mean time to go colourless. Now, to change this into a rate, I hinted at the equation earlier, we'll use rate is 1,000 divided by time, and that will give us our rate in per seconds, which you sometimes might see as s seconds to the -1. So the independent variable for this practical is temperature. The dependent variable is the rate of decay, and important control variables include the volume of lipase, the volume of milk, and the volume of sodium carbonate. It's unlikely that you'll get the mark in the exam if you simply say the amount of lipase or the amount of milk, so do make sure you're using the word volume when you're talking about your control variables.
In paper 2, you need to remind yourselves of the topics of homeostasis, which is topic 5, inheritance, variation and evolution, which is topic 6, and ecology, which is topic 7. So let's look at the main parts of all those 3. So homeostasis and response, first of all. Homeostasis means maintaining a constant internal environment. This is important to maintain the optimal, which is the best conditions for enzymes. So enzymes working in your body, for example, work best at 37 degrees C. So we'd want to maintain that temperature at exactly 37 degrees C. Not only do we want to keep body temperature the same, but we'll also want to keep things such as our blood glucose, so our blood sugar levels, and our water levels within our blood and within our body cells at constant levels. In your exam, a common question is asking you to compare nerve and hormone responses, because to maintain a constant internal environment, you might be looking at a nervous response to do this, or you might be looking at a hormonal response. So I want you to concentrate on the types of words that you need to use in a compare answer. So let's have a look at this first line here. Nerve responses are faster than hormone responses. If you're asked to compare, you must use comparative words. You can't just say nerve responses are fast. You have to make sure you're comparing it to hormones. Hormone responses are longer lasting than nerve responses. Nerves act on a precise area, whereas hormones act in a more general way. If you can't use a comparative word, use the word whereas to link your sentence. And this one, hormone responses are chemical signals, whereas nerves are electrical signals. So a common question asking you to compare nerve and hormone responses. Let's delve into a little bit more detail about nerve responses now. So it starts off with a stimulus. Now that is any change in the environment. It could be a chemical stimulus, it could be light, it could be sound, or pain, for example. Something to do with your senses. The first stage in the nerve response is the receptor. So you have receptor cells in your body that detect a change in the environment. And these are sent along the first neuron, which is the sensory neuron. So think to yourself, if you're sensing something, you're using the sensory neuron first. This goes to your central nervous system that coordinates the response. And then it goes to your motor neuron that is going to do something at the end of the pathway. And that motor neuron sends a signal to an effector, and we'll talk about what those can be in a minute. And that brings about some kind of response. So that's the general pathway that you need to remember. Let's say, for example, that someone was throwing something at you. The stimulus for that would be light reflecting off of the object. You'd detect that by light receptor cells in your eye. You'd send the message along the sensory neuron to the central nervous system, which comprises of the brain and the spinal cord. And then you'll go to a motor neuron. An effector in this case would be muscle, and your muscle would contract and move, and that might either to be catch the ball or move out the way, whichever it is. Now in this case I decided to concentrate on an effector being a muscle. There's two things that an effector can be. It can be a muscle or it can be a gland. So interesting link between the nerve and the hormone pathway, in that actually a nervous response can result in a gland being used to release a hormone. And where this comes into play is normally the adrenal gland that will release adrenaline in a very, very quick response, okay, a fight or flight response. Now, when those neurons are sending the signal along, they might have to pass the message from one neuron to another. And when they do that, they have to pass the signal across a synapse. A synapse is a gap between two neurons. So this here is the synapse. And the message within the nerve is a very quick electrical message. So either side here, the message will be transported in an electrical way. And in between, there is a chemical message. Okay, so as the nerve is carrying the signal along here, it's electrical, it gets to the end, chemicals are transmitted, which slows the whole process down slightly, and then it gets transferred onto the next neuron. So the more synapses that you have, the slower the response will be. And in the exam, they might talk about using the equation speed equals distance over time in relation to how quick nerve responses are, but they'll just expect you to know that anyway from Key Stage 3. So reflex response then. Sometimes you have a very, very quick response and it doesn't go to the brain to be coordinated. So normally our central nervous system is means that the signals go up the spinal cord to the brain and that coordinates the response. That's the coordinator. However, in this case, the message just nips across the relay neuron instead, which is in the spinal cord. So you might see a diagram that looks a little bit complicated like this. All this is showing is this part of the spinal cord. And here we have our stimulus here. We're getting stung by the bee, and our pain receptors are detecting that, sending the message along the first neuron, which is a sensory neuron. Here we've got a synapse, and in the middle, this neuron here is the one called the relay neuron. You might be asked to annotate a diagram like this in the exam and label the neurons. So rather than going up to the brain, it doesn't do that. It whips across the relay neuron and goes straight to the motor neuron. And again, the effector might be a muscle or a gland. In this case, a muscle which will contract. So moving on to the hormones then. Hormone responses. Hormones are secreted by glands in the endocrine system. Okay, so the endocrine system is a system of glands. The most important gland is the pituitary gland, because we call that the master gland, because that can switch on and off other glands by releasing hormones. So this one's the master gland. Hormones that are released like this include hormones such as LH and FSH. The thyroid gland is in your neck. That produces thyroxine, which controls your metabolism. The adrenal glands are just above the kidneys there. That releases adrenaline. The pancreas releases many hormones, and make sure you're talking about hormones and not enzymes when you're talking about the pancreas in this paper. So hormones such as insulin, for example. The ovaries produce estrogen and progesterone, and the testes in males produce testosterone. So you might be asked to label a diagram or say what is released by each of these glands, remembering that it is the endocrine system. So as we said, the response in the hormones is a lot slower. One thing that they can do is be involved in controlling blood glucose. And for that, it's the pancreas here that's the important gland for controlling blood glucose. And we said that was an important thing to control for homeostasis. So if your blood glucose is too high, insulin is released by the pancreas. Okay, so insulin is the hormone. It's released by the pancreas into the blood. All hormones are chemical messages or chemical messengers. They're carrying the message. So chemical messengers that travel in the blood. So they're dissolved in the blood plasma, and that's how they travel around. When insulin is released by the pancreas, that causes excess glucose in your blood to move from the blood into your liver and your muscle cells. So it takes it out of the blood for you, and there it is stored as glycagon, because glucose is soluble, so you can't store anything just as glucose. You have to convert it into a larger molecule like glycagon, which happens in the liver and the muscle cells. So for foundation, that's all you need to know on the blood glucose. For higher tier, I'll go into it in a little bit more detail later on. So linking into this, we've got two different types of diabetes, which is all concerned around this hormone, insulin. So with type 1 diabetes, that's the one that you inherit. The pancreas produces little or no insulin. So you may well have type 1 diabetes yourself, or know the people that are affected by type 1 diabetes. And you may well know that the treatment for that is with insulin injections or an insulin pump. So they can't produce it, so they need to inject that into their bodies to stop their blood glucose levels getting dangerously high. Type 2 diabetes then. If a person becomes resistant to their own insulin, that is when they are developing type 2 diabetes. So the doctor in this case won't give somebody insulin injections. That's not how they'll suggest that for the diabetes to be treated, because it's all to do with their lifestyle and what they're doing to have caused this type 2 diabetes. So what they'll suggest the patient should do is control the amount of carbohydrates that are going into their diet and make sure that they exercise more. And in that way, they can control their own blood glucose levels. So hopefully it won't have any other effects on their body. Okay, moving on then. Menstrual cycle. So with these menstrual cycle, the main hormones that we're talking about are FSH, and that causes the egg to mature, and LH, that causes the egg to be released. So these two hormones, I want you to remember the abbreviated ones, the FSH, the LH, they are produced in the pituitary gland. And then we've got two other hormones. We've got estrogen, which stimulates the lining of the uterus to start growing, and progesterone, that maintains the lining of the uterus. So these two are produced in the ovaries. So if you want a little visual here, think that the abbreviated words here at the top, that's the pituitary gland, and the other two longer words here down in the ovaries. So what happens? The estrogen causes the uterus lining to build up, and the progesterone helps to make sure the uterus lining is maintained before the next menstruation, when the uterus lining breaks down. On day 14, that's when there's high LH levels, because that causes the egg to be released at day 14. And before that, the follicle stimulating hormone travels down in the blood to the ovaries and stimulates an egg to mature. And obviously the woman would just want one egg per month to mature. I'll go in more detail about how these interact with each other for higher tier later on. Controlling fertility then, still using hormones. There's hormonal methods that you can use. And the hormones that we want would be estrogen, because they inhibit, which means stop, FSH. So if you haven't got any FSH, you can't mature an egg. So you're not going to get pregnant. Progesterone is the other hormone that you can use. That inhibits FSH. So the egg again can't mature and be released. So both of those are hormones that can be used. Now the progesterone also causes a sticky mucus just in front of the cervix to prevent any sperm from reaching the egg. So the progesterone does two things. Now, you could put those hormones, you could put one or the other or both of them into a pill, a patch, an implant, or injection. So these are our hormonal examples of contraception. You could use an intrauterine device, which is otherwise known as a coil as well. And there's this one is one that will be inserted into the woman's uterus and it will release hormones. Non-hormonal methods then, you've got barrier methods such as condoms, female condoms, and diaphragms. And these are otherwise known as barrier methods because they physically block the semen from going into the uterus. So you might see those as barrier methods. And alongside those, you can use spermicide to make sure you're getting the best contraception possible. And spermicide is just a chemical which kills the cells, the sperm cells. Another method you can use is copper coils, essentially copper intrauterine devices. So they actually kill the sperm rather than releasing hormones. So they kill the sperm instead. And there are methods such as permanent methods such as sterilization that you could use for the male or the female. Abstaining, which is avoiding intercourse or avoiding intercourse when you're going to be most fertile. So avoiding intercourse around that day 14 when the egg's released will help. Okay, meiosis then. Meiosis is paper 2. You would have seen mitosis in paper 1. This is the one word at GCSE where they expect you to spell it correctly, because if you spelled it wrong, you're not telling the examiner that you know whether you're talking about meiosis or mitosis. So if you remember in mitosis, that was for growth and repair. So everything from the embryo onwards to growing an individual is mitosis. However, meiosis has a very specific role, and that is to produce the sperm and the egg, so to produce the gametes. Now, in the exam, they might not well talk about humans. Okay, in humans, we have 46 chromosomes, and after meiosis, we produce either an egg with 23 chromosomes, if we're talking about a woman, or a sperm with 23 chromosomes. Okay, so we're halving the number of chromosomes that we've got, so that when they come together in fertilization, you make 46 again. But just for you to be resilient, they might not ask you about humans in the exam. They might ask you about a completely random animal, or they might even start talking about plants or something else. They'll probably keep it on animals, to be honest. But here we go. A fruit fly has 8 chromosomes. How many chromosomes would you find in a fruit fly egg? Well, it's exactly the same principle. If he's got 8 to start off with, in the egg, there'll be 4 because there's half the number of chromosomes in the gametes. So let's start and look at what meiosis is with this example of a fruit fly, because it's easier to draw 8 chromosomes than it is to draw 46. So let's look at this fruit fly. It's got 8 chromosomes. Meiosis happens in either the ovaries, so meiosis happens in my ovaries, will help you remember it, or my testes if I'm a boy. So ovary or testes. So we start off with 8 chromosomes in a body cell within the ovary or testes. Those chromosomes, just like mitosis, will have to double up. But the first thing that they do is they line up in pairs. So we've got pair 1, pair 2, pair 3, pair 4. So if this is a human, the pairs would go down to 23. And at this point, they start to switch over their DNA from chromosome to chromosome. So there's some switching, which means the chromosomes will look a little bit like this. And this switching here is what causes variation. It's what causes all of the eggs to be different and all the sperm to be different genetically. So the first division that they do is pulling these chromosomes apart after they've mixed up their DNA like so. So that's the first division, division 1. The chromosome pairs pull apart. You can see they're all slightly different now. Then we have a second division whereby these chromosomes pull apart, pull to the edges of the cell like so, and you end up with 4 gametes at the end of it. So you can see here we started with 8 at the top and now we've got 4 at the bottom. They are all different, okay, so they contain variation here. They're not the same chromosomes in each one, so they're not identical to each other. And this is the main summary of meiosis. They're to produce the gametes, which are the sex cells, which in animals are the sperm and egg. They take place in the ovaries and testes, and it causes variation in the offspring. So here we've produced 4 cells at the bottom. Sometimes we call these daughter cells. So 4 cells at the bottom. We started off with 1 cell, and there is 1, 2 cell divisions. Okay, just count these as stages. 1 division there, and a 2nd division there. Reproduction. We'll start off with fertilization first of all. Fertilization is the fusing of the nuclei of the sperm and egg. Sexual reproduction then is what involves fertilization. So when a sperm, for example in a human with its 23 chromosomes, fuses with the nuclei of an egg with 23 chromosomes, they will make a fertilized cell with 46 chromosomes. That will then start to divide by mitosis to make an embryo. So mitosis when it's growing into an embryo. Meiosis when it's to make these first of all. Sexual reproduction then, the offspring are genetically different because you've got a genetically different sperm to its father and a genetically different egg to its mother. When they combine, you're going to make different offspring. So they produce variation. And then you've got asexual reproduction, where the offspring are genetically identical to their parents, okay? They are clones of their parents. Some animals can do both asexual and sexual reproduction, and that's the same for things like plants and fungi as well. A lot of animals, however, can only do the sexual reproduction. Just to recap, in animals, the gametes, the sex cells, are the sperm and the egg. But in case they ask you in the exam, they also want you to be aware about plants, in whereby the gametes are pollen and egg. So don't talk about sperm when you're talking about plants. DNA then, moving in further into chapter 6, we need to know about DNA. So first of all, if we remember what a cell looks like, the cell has a nucleus, and that is where the DNA is kept. And the majority of the time, the DNA is loose within inside the nucleus. And at certain times, like cell division, it will make chromosomes and wrap itself up into little packages. So the DNA stands for deoxyribonucleic acid. That's the chemical that makes up DNA. It's a polymer, which means it's a long chain, and it's a double helix. So it will look something a little bit like that, where you've got one strand and then a second strand wrapped around it like so. So that's what our DNA looks like in its structure. A genome is something that we need to know about as well. You might have heard of the Genome Project, whereby they've mapped all of the genes in the human body. So the genome is the entire set of genetic material in an organism. So it could be a plant, an animal, or a fungus. If you're talking about a genome, it's all of the genetic material, so all of the DNA within an organism. So our DNA within our nucleus codes for all of our genes, okay? And these control all of our characteristics. A gene itself is just a length of DNA. So if that was our DNA, a gene would be a section here of DNA. And that codes for different characteristics like hair color or eye color and things like that. The gene is used to make an amino acid. And if you think of the opposite of the digestion module, amino acids can make proteins, because in the first paper we talked about proteins breaking down into amino acids in digestion. So you need to think about it the other way this time. Genes code for amino acids and those make proteins. So the amino acids are the short units, and they would join together to make a protein like an enzyme or a hormone with a specific shape and active site, like you will have looked at in paper 1, where we talked about proteins having a specific active site. So moving on with this chapter about DNA, we're going to look at inheritance. And first of all, we'll look at inheritance in terms of whether you're going to be a male or a female. And that's all to do with your X and Y chromosomes. And in humans, that is chromosome pair 23. So we've got 23 pairs, and the last pair determines our sex, whether we are male or female. So females have 2 X chromosomes, and males have an X and a Y chromosome. So when a female and a male reproduces, we can work out the chance of them having a male or a female. So you might be asked to complete a genetic cross like this using the sex chromosomes. So we've got the XX here, we've got XX here, and then we've got XY and XY. So we have got 50% chance of being a male and 50% chance of being a female. So just write that somewhere near the diagram to finish off the answer. 50% male and 50% female. You may well see crosses in diagrams like this as well. This tends to be called a Punnett square, this one. And you might see diagrams a little bit like this as well, whereby they're splitting up and you're having your XX here. Let's go back. You'd have your X and your X coming together, your X and your Y, your X and your Y, and your X and your X. So still the same chance, obviously 50% chance of male, 50% chance of female, but you may well see the diagrams represented like that instead. Genetic crosses then. They could talk about inheriting absolutely any characteristic when they talk to you about questions about genetic crosses. So I've just made up something random. Think about mice and think about an allele, which is a type of gene, or a form of gene, we should say. So allele is one form of a gene. That's the definition that they'd want you to write, one form of a gene. So let's say we had one form of a gene that could give you thick fur, and another form of the gene which would just give you normal thickness fur. So in this case, I've done a capital T for thick fur because this is the dominant allele. So if this is present, the animal will have thick fur, and this is recessive allele. If it's recessive, this means that if it's just present on its own, it won't have normal fur. It has to have both alleles present, like TET, lowercase, lowercase, in order for the animal to have normal fur. So let's look at some genotypes versus phenotypes. Genotypes are your genes, and phenotypes is what characteristic is expressed. So this means it's going to be a word to describe it. In this case, our characteristics are thick fur or normal. In your exam, you could be talking about blue eyes or brown eyes. You could be talking about red hair or black hair. That's your phenotype. So let's have a look for this one. Homozygous dominant. That means homo is the same, okay? So 2 of the same dominant. That would give a phenotype of thick fur in this case, because that's the dominant allele. Got homozygous recessive, which means 2 of the same, homo, recessive, so 2 lowercase letters. And this is the only opportunity for you to get a normal fur mouse. And the final combination you could have is heterozygous. So that means 1 of each, hetero, 1 of each. So you get a capital T and a lowercase t, but you need to look at which one's dominant. Dominant is thick fur. So wherever you have an allele with a capital, that means that you will express that characteristic. So in this case, because we've got a capital, that wins over. So this heterozygous will be thick fur. You could be asked to complete any genetic crosses. So for foundation, they will give you the alleles. So they could give you homozygous dominant, for example, homozygous recessive, and you would just need to complete the cross by matching up here. So we got TT, if we look across at these two, TT, TT, and then TT. So in this case, we've got 4 heterozygous, so we are going to have 100% thick fur. If we do another example where we've got heterozygous, heterozygous, when we cross those, like so, we will have 25% chance of it having normal fur. So you need to complete the cross and then write down the percentage chance for the characteristic below. If in doubt, write both of them, but in the exam they'll kind of tell you which characteristic you're talking about, whether they want you to write about thick fur or being normal. In higher tier, you will have to read some information in a question and draw your own genetic crosses from scratch. So in higher tier, they will talk to you about a situation and whether something's homozygous or heterozygous, and then ask you to complete the crosses. Polydactyly and cystic fibrosis are 2 disorders that you need to know about in this genetics topic. Polydactyly is a dominant allele. So if somebody has the allele for polydactyly, they will have this disorder. And this disorder means that somebody has extra fingers or toes. So this will happen to a few people at birth, and they'll just get removed straight at birth, their little extra digit that they've got. And yes, it is dominant, but this disorder is very rare. So if you've got the gene in your family, you'll be likely to pass it down, but it's very rare allele. So not many of the population have this allele. Cystic fibrosis, however, is a recessive disorder. That means you could have 2 parents that carried the allele. So let's just do capital B for not cystic fibrosis and little b for having cystic fibrosis, like so. If those 2 parents were going to have a baby, they themselves would not have cystic fibrosis because they've got a dominant allele for not having the disorder. However, if those 2 people reproduce and had a baby, because of the genetic cross, which we did a heterozygous one before, you would end up with 25% chance of their child having cystic fibrosis. So sometimes 2 parents have a child and they have no idea that they are carriers of this recessive allele. It means they're carrying it, but they're not expressing cystic fibrosis. So it's a recessive one. It affects cell membranes. So it disrupts the function of cell membranes. So it causes sticky mucus to build up in air passages and the pancreas as well. Now, with these genetic disorders, there's some recent technology that has suggested we might be able to do some embryo screening. So an embryo is just a small ball of cells right at the start of life. So just after the sperm has met the egg and made a fertilized cell, that will divide by mitosis and produce an embryo. And the idea is that parents could have the option, if they know that there's a history of a genetic disorder in their family, they could have the option where they produce a child by fusing a sperm and the egg together. But at the embryo stage, a doctor could remove some of the cells and screen them, which means check them for genetic disorders. And then the idea would be that they would pick an embryo that doesn't have a particular genetic disorder in. So there's lots of moral implications with this. Some people say that you shouldn't be able to choose what child you have, but obviously there's positives with you bearing a healthy child, and later down the line, even though it's expensive, it might reduce the costs on the NHS for further treatments if somebody did have the disorder. On to the evolution topic. With every evolution question, what I would like you to do is almost ignore what animal they've got in front of you or what plant they've got in front of you. Don't be put off if they start talking about axolotls or limpets or lichen or things that you've never heard of before. Think about the framework for evolution by natural selection and try and get these answers in no matter what. So the idea that there's natural variation in the population anyway, and a mutation, which is a change in the DNA, causes a change in phenotype. If you remember, that's what the animal looks like. So if you can't remember phenotype, it's what the animal or plant looks like. So individuals best adapted to the environment will then survive. So get the words best adapted and survive in there. They will reproduce and they will pass on their genes to the next generation. So absolutely any question, regardless of the animal or the plant that they're talking about, if they're talking about evolution by natural selection, you need to get those steps in. Now, when they're talking about a mutation causing a change in what the animal looks like, then you can link it to the question. So, for example, if a mutation caused a snake to be green, that might make it camouflage in the grass better. If a mutation caused a marine iguana to be black, again, that would cause it to camouflage better into the rocks. Or it could be a mutation that caused something to have wings and allowed it to fly, or whatever it is. So the last thing to do is relate it to the question itself. Speciation then, leading on from natural selection. If you have isolation, so two populations, let's take some sheep, for example. Just by nature, some of those sheep will be naturally fluffier than the other ones. So let's do some normal sheep and some really fluffy sheep, like so. If those sheep were to be separated, they could be separated by a mountain range or a river or a lake, for example. That suggests a river, large river, became between two sheep populations. We might have one on this side, like so, and one on the other side. Now, at the moment, these sheep can still interbreed with each other and make offspring. But because of the isolation, because they can't get across the river, these might end up on two different islands and they might gradually drift away from each other. When they do, they might find themselves in slightly different climates. It could be that there's different food on the island for them to eat, and they might adapt and evolve to eat those foods. Or it could be that there's different environmental differences. For example, on this island, let's say that they drifted off to somewhere that was very sunny. If you think to yourself about evolution by natural selection, think to yourself which sheep would survive best on this island. Well, probably this sheep here is the one that is best adapted. So that will survive, reproduce, pass on its genes to the next generation. So gradually over thousands of years, these thick-furred sheep would die out and you'd have the lesser, the normal sheep or the non-thick-furred sheep surviving and reproducing and being more common in the population. So you could get a change where actually when these sheep were to meet again, it could be that they can't breed anymore because they're so different that they're actually now a new species. So if they're talking about making a new species, talk about natural selection that we talked about previously, but add in the word isolation. And if you end up making a new species, by definition, if two individuals are classed as different species, if they can't reproduce and produce fertile offspring. That's a key definition for what makes a different species. So if these came back together a few thousand years later and they were very, very different looking, but they could reproduce and make fertile offspring, then they would still be the same species. But if they came together and they changed so much that they couldn't reproduce, they might be able to make babies, but those babies can't make their own babies, so they're actually not fertile. Then we would call them two completely different species. Selective breeding then, a couple more things. So we'll talk about selective breeding. That's when you are choosing the animals that you want to breed together or choosing the plants that you want to breed together. So you're playing with nature, you're playing with the natural selection cycle, and you're not letting nature choose those animals that are best adapted. You are choosing them for various reasons yourself. So you'd select a male and a female with desired characteristics. But for example, if you wanted a cow to produce lots of milk, you'd need to talk about not selecting a male that would produce lots of milk, but with this case, a male whose daughters would produce lots of milk. So in this specific example, you'd either choose the male and the female with a desired characteristic, or where appropriate, whose offspring have a desired characteristic. Okay, so if you're talking about cows and milk, obviously a male is not going to produce milk. So you'd choose a male that has daughters that produce lots of milk and breed that with a female. You breed these individuals together, and from the offspring, you select those individuals with your desired characteristic. It could be producing more milk or more meat or whatever it is. And then you breed those. And then you continue this process for many, many generations. So always picking your individuals that you want to breed and breeding them together. So you might want to do this for a higher milk yield. So yield is how much you get. So getting more milk from cows, and that's what we've done over several years and several generations of cows. You might want to get a higher meat yield. So we've done that with things like chickens and continually bred them until they're producing us more meat. Or in the plant world, maybe something that's got bigger flowers or better flowers instead. If you don't want to do selective breeding, this is something we've done for hundreds of years, but more recently, technology has allowed us to do something called genetic engineering. And with genetic engineering, you take a gene from one organism and insert it into another. Now, you can really let your imagination go wild with this. So there are stories of people taking a fluorescent protein out of jellyfish, for example, and inserting it into cats to make glow-in-the-dark cats. You can literally take any gene from one animal or plant and insert it into the genome of another one. Now, you need to do that at a very early stage, like when it's an embryo. You can't just stick it into an adult animal and expect it to change. You might want to do this for crops so that they're resistant to herbicides, for example. Take a herbicide-resistant gene and put it into your wheat crop. Crops that are resistant to disease. Perhaps crops with increased yield. You can tell that I'm talking all about plants because the mad example of the fluorescent protein in a cat is not really a moral one. So we tend to do this on plants rather than animals. And bacteria, for example, to produce insulin, you can take the insulin gene out of a human and put it into bacteria. And I'll go into this in more detail for higher tier later on. These, when they are genetically engineered, are called GM crops. Okay, genetically modified crops are those that have been genetically engineered. So classification. Here's a little bit of a rude rhyme to help you remember classification. So king penguins crap on French girls' skirts. Okay, so that mnemonic there will help you remember how we classify animals. So, and plants and fungi and everything else. King stands for kingdom, P for phylum, C for class, order, family, genus, species. So this classification system here is called the Linnaean classification system, and it was developed by somebody called Carl Linnaeus. So he came up with this system, and all of the animals and plants and fungi and bacteria that he could see, he put them in this system. However, another Carl came, okay, it's one for the Carls this, another Carl came, and he said, I think there should be something above kingdom. So a guy called Carl Woese, this time, W-O-E-S-E, came up with the three-domain system. So he said, above kingdom, we should have three domains called eukaryota, where all the eukaryotes are, so plants and animals. All the bacteria should be in a separate one. And this is your true bacteria, what you know as those kind of single-celled things. And then archaea is the third domain. Now, these two are very similar because they are actually both bacteria. But this one you call the true bacteria, and this one is very primitive bacteria. These are where your extremophiles are. Extremophiles are bacteria that live in extreme environments like high pressure, high temperature, high salinity. So he added these domains. And then under eukaryota, you still have the kingdom, phylum, class, etc. But this is now on top. Now, key is they like questions about why you change the model and why you change the system. And it's all to do with new evidence coming to light. Okay, so if they ask you about why the system's changed, talk about new evidence. So the advances in microscopes and things like that let us look at the subcellular parts of animals and allowed us to compare whether they had nuclei, whether they had ribosomes and mitochondria and things like that, which then allowed us to separate them into other groups. And it may well change in the future. Right, last thing on this then is the binomial name. The binomial name, bi for two, like bicycle, nomial name, comprises of the genus and the species. So, for example, for humans, our genus is Homo and the species Sapiens. So that would be our binomial name. Take a barn owl, for example. You don't need to remember these examples, but if they give you the binomial name as a barn owl of Tyto alba, they might say to you, what's the genus? So the first word is the genus, so you'd write Tyto, and for reference, that word is the species. So those two together give something its binomial name. So they may well give you a table and then say, write the binomial name. So you just take genus and the species from the table and write those together. A few more bits on ecology then. Ecology topic, it starts with a lot of words. So let's start off with population. Population means that all the organisms of a single species living in a habitat. So if we talked about fish, for example, this would be a population of fish. A community is populations of different species. So you might have a fish in there, you might have a different species of fish, a much larger predatory fish, for example, and maybe some plants in there. That would be a community. An ecosystem is the interaction of living, biotic, and non-living, abiotic factors. So it could be that you're talking about the rocks that are in that system, the temperature of the water, the current of the water, and adding all the abiotic things in there as well. And two more keywords, biodiversity. Bio means living, diversity means variety. So biodiversity is the variety of living organisms on the earth. Final word, interdependence. That is the dependence of different species on each other. So some species could be dependent on each other for food, perhaps for shelter. So birds are dependent on trees for shelter, for example. Pollination, so bees depending on particular species of flowers depending on particular species of bees for pollination, for example. So we talked about abiotic and biotic factors briefly. Biotic factors are the living ones. Biology is the studying of living things. So bio means a living factor. Predators are an example of a living factor. Pathogens, so causing diseases, competition between each other animals, and food availability. So if you think about these donkeys here, they would be competing with each other perhaps for food, and this would affect their survival. Abiotic factors, non-living factors, lots to do with weather on this one. So temperature would affect that ecosystem, wind intensity, amount of rainfall, pH, salinity, which is how salty it is, and oxygen availability, for example. Have a look at what situation you're given, because if you're talking about oxygen availability, you're only talking about aquatic environments. So if we're talking about the water here, we'd be talking about things like how salty it is and oxygen and temperature, but obviously we wouldn't be talking about wind with that one so much. Adaptations then. They could ask you again about adaptations of any animal or plant. So just look carefully and use your knowledge of what's in front of you to think about things that are behavioural, structural, and functional. Behavioural is how the animal behaves. Structure is what it looks like, what its structure's like. And functional is anything that happens inside the body. So anything to do with its metabolism, or whether it respires, or the processes that happen inside the body. So for example, if we take this bat here, we could talk about behavioural. It hunts at night to catch its prey. It roosts in groups, for example, for insulation. You don't need to get this specific example down, but I'm just showing you how you could answer this type of question. Structural, that's what it looks like. So large wings for flying, thick fur for insulation, brown fur for camouflage. And functional might be something like the fact that it hibernates in winter. Internally, it lowers its body temperature. So whatever animal or plant you're given, try and think about, could be a 6 marker, for example, try and think about how it behaves, the structure of it, and things that happen inside of it. Water cycle then, nice and easy. It just needs to it needs for you to recall 4 words. The first word is evaporation. So from seas and lakes and things like that, the water will evaporate. It will then move up into the sky, where it will condense together to form clouds. And then at a certain time, you will get the rainfall, which we call precipitation. So don't just talk about raining, use this posh word here, precipitation, instead. And the final factor is to do with these trees. From paper 1, you'll know that trees and plants also lose water, and that process is called transpiration. So 4 things to consider about the water cycle. We also cycle carbon. Now, carbon is a chemical that is in an element, that is in lots of plants and animals, the majority of plants and animals. So we just need to talk about here about the way this is cycled around our planet. So one way is feeding. So if this cow here eats the grass, then the carbon is being transferred from the grass into the cow. Photosynthesis. So if we think about carbon being in carbon dioxide, then the plants will take in carbon dioxide from the air, and that's part of the carbon cycle, and they'll obviously release oxygen. They'll produce glucose in that process. Glucose has carbon in it, C6H12O6. So when the animal eats the plant, then they'll be taking that carbon up as well. Respiration, both animals and plants respire. And when they do that, they will be releasing CO2 to the atmosphere. Combustion, if we've got fuels inside the tractor here, for example, in factories, cars, if we're burning fossil fuels, we're releasing CO2 into the atmosphere. And decay. Now, this is something I do want to highlight, because this is a common theme in the exam, talking about decay. Decay is caused by microorganisms, so small organisms, things like bacteria, etc. Now, microorganisms are involved in this cycle as well, because they also respire. Just because they're really small, don't count them out. They respire and release carbon dioxide. So when the dead plants and the dead animals are decaying in the soil, the microorganisms are contributing because they're respiring when they decay them, and they release CO2. In global warming, I want you to remember, from the sun, short wave radiation is released. This comes up in the physics topic as well. Short wave radiation is released. That goes through our blanket of greenhouse gases in our atmosphere, but it is reflected back after it's reabsorbed and then re-emitted from objects on the Earth as long wave radiation. But the trouble is, the long wave radiation finds it very difficult to get through this layer of greenhouse gases. So what it does is it's reflected back down to the Earth, and that's what causes it to warm up. And the consequences of that you should be aware of from the news, things like flooding and extreme weather and hurricanes and things like that. Waste can come from a number of different ways, and it's mainly caused by the fact that our human population is rising at a very, very fast rate. So it's like that for thousand years, and then suddenly we are increasing at a very fast rate. We're producing toxic chemicals, solid particles in the smoke that we release, sulfur dioxide in the smoke that can cause acid rain, nuclear waste, and sewage. And we're having trouble at the moment trying to get rid of these and store these so that we're not polluting and damaging the planet. Now, one of the ways that I want to dwell on, because it does come up sometimes, even though it's not made obvious in your spec, is eutrophication. So you can see here we've got a really green lake, and that green lake is covered in algae. So what happens is fertilizer can run off from the farmer's land. So lots of nutrients in the soil here going into the lake or the river. The plants and algae love those nutrients. So they use the nutrients from the fertilizer and they bloom and they multiply and they cover the lake. When they cover the lake, this reduces the sunlight that can get in. And this means that the plants in the water die because they can't do photosynthesis anymore, so they die off. Again, relating to microorganisms now, the microorganisms that are in the lake decay the dead plants. And the link that I made before, these microorganisms respire. And when they do that, they use up the oxygen in the lake. And this can cause lots of larger animals to die, like the larger fish and things will now die because the oxygen levels get so low. Higher tier contents. You need to be able to construct a Punnett cross from scratch. So that's going back to crossing heterozygous or homozygous dominant or whatever it is and drawing a Punnett square from the information given. We're not going to dwell on that too much, but just read the question carefully in the exam. We're going to look at hormone level interactions, glucagon, another hormone, more hormones again, adrenaline and thyroxine, IVF, and a little bit more detail on genetic engineering. So hopefully you would have had a nice summary of the foundation step of 4, and remember to concentrate on these bits because this is where your paper might stand out a little bit different from the others. So negative feedback for separate and combined higher, this word comes up a lot. Basically, it means if something gets a little bit too high, a mechanism will come in to bring it low, and if it gets too low, a mechanism will come in and bring it up again. So it's very difficult to keep, for example, your blood glucose levels exactly normal all of the time. So it will go a little bit high, and then the hormone insulin will bring it back down again, but it then might go a little bit low. And another hormone that you need to be aware of is glucagon, which will then cause that to rise again. They could well say to you, give an example of a negative feedback cycle in the body. So if you want to remember one, you could remember the insulin and glucagon one. So we said before, if the blood glucose level is too high, insulin is released by the pancreas, glucose moves from the blood into the liver and muscle cells, and the glucose is stored as glycogen in the liver. Now, you need to know the extra bit in what happens when the blood glucose level is too low. So here we've got a separate hormone. The pancreas releases now glucagon, and this hormone travels in the blood and it converts glycogen in the liver and the muscles back into glucose. And that glucose is then released back into the blood. So this part of it is higher tier only because this links into the idea of a negative feedback system. Hormone interactions, again, higher tier only. So the hormones we said at the top were FSH and LH, and at the bottom we had estrogen and progesterone. Now we're going to talk starting at FSH. FSH is produced in the pituitary gland. It travels in the blood to the ovaries, and what it does is it stimulates the ovaries to start making estrogen, as well as obviously causing the egg to mature. So it stimulates the ovaries to make estrogen. The estrogen then travels through the blood to the pituitary gland. So I'll just put O for estrogen. And that actually says stop to the FSH. So the estrogen inhibits the FSH because you only want one egg to mature. If you had lots of FSH, you'd have lots of egg maturing, which is not what you want in the menstrual cycle. So the estrogen inhibits the FSH, and at the same time, it stimulates LH. And LH is produced in the pituitary gland, that travels down to the ovaries, and then that causes the egg to be released. Okay, so you need to know about estrogen and FSH and LH and how they are interacting to control what happens in the menstrual cycle. Adrenaline and thyroxine then, two more hormones that you need to know in more detail. Adrenaline, produced in the adrenal glands, and it's for a fight or flight response. So this is often at the end of a reflex pathway as well, adrenaline, to start it off with. And it increases the supply of oxygen and glucose to cells in the brain so that you can consciously think about what you're doing and make quick decisions, and your muscles so that you can move out the way. So if you're scared, your response may well be to fight or may well be flight, to run away. But either or, you get an increased supply of oxygen and glucose to your cells in the brain and muscles for obviously respiration. Thyroxine is produced by the thyroid gland, and that controls your basal metabolic rate, so your resting metabolic rate. And the levels of thyroxine are controlled by TSH, thyroid stimulating hormone. And this hormone can control the levels of thyroxine in your body, because you need to control your basal metabolic rate. And again, you will have a negative feedback cycle. So high thyroxine levels, you might have a cycle that looks like this again, where it goes high and then back to normal and then low again and then back up again. So if it gets too high, you want to stop the thyroid, so you want to inhibit the TSH. And if they get too low, the thyroxine levels, you want to stimulate the TSH to go to the thyroid and produce more thyroxine. So at this point, you've got the TSH inhibited, and at this point, you've got the TSH released to control the levels of thyroxine in the blood. So this could be another example of negative feedback that they talk about. IVF then stands for in vitro fertilization, and it's when a man and a woman decide to have a baby. If they're finding it difficult to have a baby and can't conceive naturally, they might choose the route of IVF, whereby the man provides sperm and the woman provides an egg, and the doctor will fertilize that under a microscope. So the woman takes regular injections of FSH and LH because these are the hormones that mature the egg and release the egg. So if she takes lots of injections, that means she can take multiple eggs, not just one, to be matured and then released. And then the doctor can do an operation to retrieve those eggs. So she may have a dozen or so eggs at the end of the process. The man provides a sperm sample, and the scientist injects the viable sperm, so the best swimming sperm, into the egg. The fertilized egg then divides to form an embryo, like we've discussed before, divides by mitosis to form an embryo. And then normally nowadays, only 2 viable embryo are inserted into the woman's uterus. That's why it's common for IVF patients to have twins, because if 2 embryos work, they will have twins. Previously, they might have put up to 8 in there, but now it's getting more successful. They're only normally putting 2 in there. You might be talking about IVF. It's stressful for the woman. She has to take lots of injections and has to have an operation. It's not always successful either, and it costs a lot of money. So genetic engineering, a little bit more detail than we looked at it for foundation. Here we've got the idea that we said before, that you take a gene from one organism and insert it into another. This, for example, could be the human gene for producing insulin, And you'd want to cut out that gene using enzymes. And those enzymes are normally called restriction enzymes. If you forget that word, just write about enzymes. But enzymes cut the gene for insulin. And this is just one example. They could talk about a gene for herbicide resistance or another hormone that we need to produce, for example, TSH or something like that. They could talk about producing TSH or whatever it is. In this case, we use a bacterial cell because they divide very rapidly, and they have a ring of DNA called a plasmid. Now that plasmid, in this circumstance, because it's going to carry our gene that we want, we call that a vector. Okay, so the plasmid in this case is a vector. Sometimes people use viruses, but in this case we're using a plasmid instead. So you need to use the same restriction enzymes to cut a bit of the plasmid away, and then combine those together using another enzyme, which is called ligase. Again, if you don't remember that, just talk about enzymes using it and a different enzyme. You then put that back into an empty bacterial cell, and that bacteria would then replicate, and produce the plasmid containing the gene for insulin. So what the scientists can do is then allow this to replicate at a rapid rate and then purify it so that you end up with a nice bottle of insulin produced by that gene there that you can then sell for insulin injections for type 1 diabetes, for example. So they might talk about any situation in the exam. They might talk about genetically modifying a crop or something like that. So don't be put off by what situation they're talking about. They may well talk about bacteria and insulin, or they may talk about something instead. Separate scientists then, this looks like a busy page, doesn't it? There is a lot of separate science only content. So where are we going to start off with? We're going to briefly go through these two required practicals. They're in more detail in the required practical video if you want to watch them, but for separates, plant growth and decay. So we'll talk about plant growth first and decay a bit later on. So with plant growth, you're just talking about changing one factor, like it could be light intensity or light direction, and see how that affects plant growth. So nice and easy practical for this one. You'd have the light at different distances. And the important thing is control variables. So same number of seeds per dish, power of the lamp in this example specifically, the type of seed that you're using, the volume of water, and temperature, things like that to keep constant. And you might simply measure the height of the plant and do some biological drawings to show the height of the plant. And you might get something like this. The higher the light intensity, the higher the plants grew. Or you could change the position of the light source, because we're going to lead on to talk about plant hormones and phototropism. So the position of the light source. So you could have one coming from the left, from the right, from the top. Again, keeping all the control variables, the important control variables the same, and doing biological drawings and perhaps measuring, drawing to show the direction of light, or you may measure height and direction. It's up to you. The angle of the Petri dish, you may well have some crazy setup that they've got in the exam where they're changing the angle of the Petri dish for measuring plant growth. So this again involves plant hormones and auxins that we'll talk about in a minute. So you might put the plant Petri dishes at different angles, this time keeping the light source the same number of seeds again, the same type of seeds, etc. And this time you might be looking at measuring the direction of the plant growth. And that might be simply for an observation. We don't necessarily have to measure anything. We might just say it grows up, down, left, or right. And you would have results looking a little bit like that. So that's the first required practical. And it's all controlled by plant hormones. And what I want you to remember is this plant hormone with growth is auxins. So auxins stimulate the growth in the shoots. So S for stimulate, S for shoots. Auxins are in the tips of the roots and the shoots. So if this was our plant here with our shoot and our roots, like so, coming out of the ground, the auxin would be here and here. Now, it does two different things. If it's in the shoot, it will stimulate growth, okay, up here, cause more growth. And in the roots, wherever the auxin is, it actually stops the growth. It inhibits it. So if we start with a seed that is in the soil, it will have a shoot and a root. Now, let's first talk about geotropism or gravitropism, which is the response to gravity. Due to gravity, the auxins will lie on the bottom of the shoot and on the bottom of the root, like so. Because of our rules up here, in the shoot, it stimulates growth, which means these cells here will elongate and the shoot will grow upwards. On the root, it inhibits growth on this side, so these cells will grow more and elongate, and the root will grow downwards. So two different things depending on whether it's the root or the shoot. When we talk about this shoot growing up, let's say the light source was over here. We can now talk about phototropism. This doesn't affect the roots because the roots aren't responding to light, but the shoots are. Now, for as long as there are equal distributions of auxin either side, the plant will grow upwards. But in response to light, the auxins move to the shady side of the plant. So they'd move across to this side. This would cause more growth on this side, causing the shoot to bend towards the light. So the important words to be talking about is unequal distributions of auxins. So unequal distribution of auxins, that's the hormone that we're talking about, causing unequal growth rates. Try and get those keywords in when you're talking about plant hormones. Now, some plant hormones are used in industry. So let's go through a couple of those. You've got gibberellins, first of all. G for gibberellins, G for germination. So gibberellins are hormones that are involved in germinating seeds. So they can end seed dormancy. So if you buy a packet of seeds like that, you have dormant seeds, not ready growing, but the hormone gibberellin can promote those seeds to start growing. They can promote flowering and make bigger fruits as well. So industry might try and use these to perhaps increase the yield of their fruit crops or produce better flowers, for example. You've got ethene. Ethene is used in fruit ripening. So some fruit that you buy might be packaged, and the company might have put some ethene in there in the packaging to control the ripening of the fruit during transport and storage, or to prevent ripening, they might have something to remove any ethene that's released. And auxins, because they are involved in growth, they can be used in weed killers because they cause uncontrolled growth in like broadleaves, so weeds like this that are sticking out the ground. They will leave the grass, for example, that'll be fine, but the auxin will cause uncontrolled growth of these, which will cause them to die out. It can be used as rooting powders and promoting growth in tissue cultures, which we'll come to next. So keeping on the theme of plants, first of all, in the cloning topic, you might be asked about cloning plants. Well, quite simply, you can take a cutting of a plant, so perhaps a leaf and a bit of stem, or even just a leaf, off of a plant, and you could grow that in soil. If you dip that in auxins, that will promote the growth of roots. So we'll dip it in some rooting powder there, and that will cause our roots to grow. And we will get an identical clone plant. For rare plants, we don't really want to be chopping off leaves or bits of stem. So instead we can do tissue culture, whereas instead we'd scrape a few cells off of the plant and we'd grow that in nutrients in an agar dish, first of all. So you just put a few bits of the tissue in there instead. And then when it was older, you could then grow it in soil and separate the plants up and divide them up to try and replicate them. Cloning for animals then, you could do embryo transplants. So if a farmer had two prize high meat producing cows that they wanted to breed together, they might produce an embryo, which is going to be a desired animal. And at this stage, they might hedge their bets and assume that this is going to be a really good animal, and they might take the cells from the embryo and just allow them to divide again to make identical embryos. Okay, so take a few cells out at the early stage, stimulate them to divide, and you will have 3 identical cattle at the end of it, for example. However, you might want to do embryo screening before that, because that's quite brave to do. You may well end up with 3 genetically identical cattle with a genetic disorder, for example, that is going to be absolutely worthless to you as a farmer if you can't use it in the meat industry. So that's quite an early stage to do things. The other more recent one is adult cell cloning. So that's when we can actually clone a whole animal. So if we had our prize sheep here, for example, you could take out one of her adult cells, like a skin cell or a muscle cell, anything containing her DNA, and you would remove the nucleus. So you've got her DNA in there, or his DNA, whatever you wanted. We've got another sheep here that is just going to be our egg donor. Okay, so this female sheep is going to provide us with an egg, but we don't want her DNA in it. We want to take that out, so we'll remove the DNA. So we've got an empty egg cell from a donor and let's call this Dolly, because this is what the sheep was actually done on, Dolly. And we've got the nucleus from Dolly. We combine those 2 together, give it an electric shock, and it will start dividing, into an embryo. And this will continue to divide and will make an identical Dolly. We'll need to use a surrogate, which is just a female that is going to take bear the child, so it's going to have the embryo implanted into the uterus. So let's add in here a surrogate mother to make the diagram more complicated. So we put the embryo into the surrogate mother, and then she will then give birth to identical Dolly. Okay, so an identical sheep that we started off over here. Basically, you can think about it like therapeutic cloning from paper 1. But rather than leaving it at the embryo stage, we're going to implant it in a surrogate mother and allow it to develop into a full animal. So that's the extra little bit. Okay, so decay required practical. You could look, for example, at how temperature affects decay. They could ask you about pH or something else in the exam, but here we're going to concentrate on temperature. We're using an enzyme here, so thinking back to paper 1, lipase is an enzyme that breaks down fats or lipids into fatty acids and glycerol. So we start off with 5 centimetres cubed of lipase in one test tube, and in another we add some milk. That is going to contain our fat. We add a few drops of phenolphthalein, which is our indicator. That's pink in alkali and colourless in an acid, and some sodium carbonate solution. This solution is going to make sure that our solution starts off as an alkali, so it's going to start off pink. In the exam, they might use a different indicator and they'll give you a clue about the colour change, so don't worry if they give you a completely different indicator. So we put all of that into one test tube, and we've got the lipase in another, and we need to put them into the water bath so that they stabilise. We might have one at 30 degrees, for example, 40 degrees and 50 degrees. Now, what we need to do is we then need to take, so it starts off pink, we then need to take 1 centimetres cubed of lipase, once it's made up to temperature, transfer that into the other test tube, and time how long it takes to go colourless. Okay, time to go colourless, and we'd repeat that again for the same temperature, and we'd repeat it for the different water baths. Now, the reason it's going colourless is because the lipase is breaking down the fats in the milk into fatty acids. Okay, so it's changing the conditions. We started off with alkali. Don't worry about the glycerol as well. We're concentrating on the fatty acids, but it breaks it into the fatty acids, making it acidic. Again, because it's a colour change, if we're calculating the rate of decay, you might be using the equation rate equals 1,000 divided by time. So that's the other required practical that they might talk to you about. Moving on to decay, Again, we're talking about the little guys. Hopefully these will feature quite a lot in the exam. Microorganisms. Sorry, excuse my writing. Microorganisms. And we're talking about before about them respiring and releasing CO2. This time we're talking about them respiring anaerobically in a biogas generator. So one of the problems we had with waste before was a lot of sewage. So this is one way we can get rid of sewage. So we'd put the sewage into the inlet pipe here, and in here we'd have some microorganisms. And we'd have microorganisms in anaerobic conditions. We'll come back to the meaning of batch and continuous in a second, but we'd make sure that they were in anaerobic conditions, because if they are in anaerobic conditions, when we got our sewage in here, it releases biogas, which is mainly methane. That is a byproduct of the anaerobic decay in here. And then, as a useful product, we get the digested material out here that makes really good fertilizer. So we can store that and use it again on the farm. Some farmers have small biogas generators, and they are batch generators on their farm, which means that they themselves shovel the waste from the farm in here, and that will produce biogas, produce methane, which they can use to generate electricity, or they'll sell it on, and to produce fertilizer. So this is just stop and start. So they put one batch in, get the methane out, stop it again, and then start the whole process. Or in a larger scale, industrial scale, you could have continuous, where all the sewage is coming in from pipes, continually going into the biogas generator, costing a lot more to set up, but making a lot more biogas in the first place. So they might ask you about choosing, evaluating maybe a particular generator for a particular situation. DNA then, DNA in more detail. Before we said it was deoxyribonucleic acid, and it was a polymer and a double helix, and we pretty much stopped there. But you guys need to know more detail about the structure of DNA, and then we'll link into how proteins are made. So this is the structure of DNA. You've got a sugar phosphate backbone. So you've got the phosphate, the sugar here, creating a backbone alongside. And you've got bases in the middle. Those bases can be A, T, C, or G. So A, T, C, or G. The word nucleotide might come up. This consists of this triplet here. Okay, a base, a sugar, and a phosphate. One of those is called a nucleotide. So if we had our DNA as a helix like so, we've basically unzipped this and we're showing both strands here, one strand either side. These bases complement each other. So A is a complementary pair with T, and C and G are a complementary base pair. You can't have A and G together here, or T and C together here. In terms of making proteins then, within the nucleus of a cell, you have the DNA, but that is too large a molecule. It's a very large polymer. It's too large to get out of the nucleus. So if you want to make a protein, if you remember, they're made at the ribosomes in the cell. So you have to get the DNA somehow from the nucleus to the ribosome to make the protein. So what you do is you actually copy your DNA into a strand called mRNA, messenger RNA. And this carries a copy of your DNA code. This travels to your ribosome, and this will read the code 3 bases at a time, like so. Every 3 bases will code for an amino acid. We said before that amino acids will then make proteins. In the body, there are 20 different amino acids that you can make through this process, and you'll fold them up to in different shapes to make different proteins. So the mRNA will go into the ribosome like so and will be read. And the carrier molecule, which you may well have been taught as tRNA, the transfer RNA molecule, but in the exam you can just call it a carrier molecule. That brings the correct amino acid here and puts it onto the sequence. Okay, so it would read that CGA and it would come and drop the correct amino acid there. And it would repeat that again and again until you've got a nice chain of amino acids like so, which would then fold up to make your particular shaped protein. Now, most DNA actually doesn't code for a gene. You've got non-coding DNA, which can code for turning on and off genes. So this is coding DNA, because we're actually talking about it making amino acids and proteins. But at the end of the chain, you'd have something that stops that protein when it's finished at the end. And lots of DNA is non-coding, and it doesn't code for stuff. Now, moving on to mutations in DNA, there's three different types, that you can have. An insertion mutation, a deletion, or a substitution. Now, let's clear this off here, and let's have a look back at the molecule while it's over here. If we had a deletion mutation, that would mean that we would remove one of the bases. We're deleting one of the bases. And what that would mean is that your mRNA would be read totally differently, because rather than your first amino acid being coded by CGA, it would now be coded by ACA. And it would affect the one along the next chain as well, and the next chain. So it has a knock-on effect down the chain. In a similar way, if you inserted an extra base in here, for example, we had an extra C in there. When the ribosome was reading it 3 at a time, again, it would affect the whole chain. So it would have a knock-on effect after that insertion. So inserting one is called an insertion. What's slightly different to that is if you had a substitution. Let's take this triplet here. If, for example, this base was changed for a G, and it was CGG instead, that would only affect that amino acid here, and it wouldn't affect the other amino acids. So most mutations have no effect on the protein and won't cause any harm. Only some mutations would cause a really massive change in the protein, that it would actually cause a disorder genetically. Moving on to the brain then. So the brain, there's 3 parts of the brain you need to know. The cerebral cortex, which is the big squidgy stuff. And this controls our consciousness, intelligence, language, and memory. So think of it as the big bit of the brain that's controlling all of these important things. You've got the medulla, this region of the brain, that controls unconscious activity. And if you think to yourself, it's quite close to the spinal cord, so you're kind of before you get to the real part of the brain. So think to yourself, anything unconscious doesn't involve the brain. So the medulla is the bit right at the base of the brain. And then you've got the cerebellum, which is for muscle coordination. So any damage to the back of the brain could affect your muscles. Any damage up here would affect your intelligence, language, or memory. Now, doctors that investigate problems with brains are called neuroscientists, and they might they have mapped particular regions of the brain to their function by electrically stimulating parts of the brain and scanning it. So sometimes patients that have brain disorders, they will stimulate certain parts of the brain using probes and then look on a machine to see what effect that is having on the activity of the brain. So it's telling them a lot about the different parts of the brain and what they do. But obviously, any operation with the brain can have serious consequences. A little bit on temperature regulation. If you are too hot, you're going to want to cool yourself down by sweating or vasodilation. So sweating, if sweat evaporates from your body, this has a cooling effect. Vasodilation, the blood vessels dilate, which means they get a little bit wider. They increase the blood flow to the skin, and this increases energy transferred to the surroundings. So if this is your skin, you're going to be transferring more energy to the surroundings if your blood vessels have dilated and there's more blood flow. So your hands will go red and quite swollen. The opposite, if you are too cold. Vasoconstriction, you might well have done this in PE, for example, and shivering. So the blood vessels constrict, and that reduces blood flow to the skin, so you're going to reduce the energy loss to the surroundings. And with shivering, that's when your muscles contract automatically. So when your muscles are contracting automatically, this requires respiration for contraction. And during respiration, obviously, the main purpose of that is for energy to be released. So shivering automatically means that you're going to be releasing energy in your muscle cells. This, again, is an example of a negative feedback cycle. So if you're getting too hot, you're going to sweat and vasodilate, but then you might go down into the region where you're a bit too cold, and then you'll shiver and vasoconstrict until you're regulating that body temperature around 37 degrees. Here's the kidneys then. The main purpose of the kidneys is to filter your blood to get a lot of the waste products out. And then some of the bits that it's filtered, you actually want back in the blood. So we call that selective, choosing what you want, to reabsorb, selective reabsorption. So those two things in order are what the kidney does, filter and then selectively reabsorb. So one of the things that it's going to help control is water levels. And again, this links into the topic of hormones, because the hormone that helps control that is called antidiuretic hormone. It's released by the pituitary gland, which is the master gland in the head. And again, it's part of a negative feedback cycle. So if this is our normal levels in the blood, they might get a little bit too high and they need to be controlled and brought back down. Then they might get a little bit too low and up and low, and etc., etc. So the ADH, this hormone, increases the permeability of the kidney tubules. And what this means is that more water is reabsorbed from the kidneys back into the blood. So it controls this selective reabsorption process. So if your water levels are getting too low, you're going to release the ADH, and that's going to increase your water levels in the blood. And if they're getting too high, you then want to inhibit the ADH to bring the levels of water back down again. Linking with the kidney still is the waste product urea, which is removed by the kidneys in the filtration process. And the urea is formed from protein digestion. So linking back to paper 1, when proteins are digested, they form amino acids. And too many amino acids, any ones that are in excess that we're not using for growth in our body, need to be removed. And this process happens in the liver. And we say that the amino acids are deaminated. And you don't need to know in this detail, but the amino acid has an amine group here, which is why it's called deamination. You're basically taking this bit off of the molecule. And you can see here how this is then going to form ammonia. So ammonia is formed in this process, and it is toxic. So you need to get rid of that very quickly from your body. So the ammonia is quickly converted into urea and then removed by the kidneys. So that is the process of deamination, which happens in the liver. And then the kidneys are the ones that remove the urea by filtration. The other thing before we move on to the genetics that they might ask is about the dialysis machine. So kidney patients that are having kidneys that are not filtering out the blood can be very dangerous, because if you have things like urea in there, if you have things like ammonia in there that's toxic, you can then make the urea and then you need to get that out of your body. So if your kidneys are not working, you might need to go onto a dialysis machine. So have a look in a little bit more detail into that for your exam ready tomorrow. So Mendel. In brief, we're going to go through the evolutionary scientists. I don't want to spend about 10 more minutes on this video, so we'll go through a little bit of the evolutionary stuff before we do a little bit of ecology. So Mendel, he crossed pea plants and looked at the characteristics of the offspring. So he had a really tall plant and a really short plant, and he crossed those two together, and his first generation of plants were all tall. Okay, so when you're talking about the first generation, that's the first offspring. And then he crossed this generation to get a second generation of plants. So he crossed those again to get a second generation, and he found that actually out of that second generation, some of them were small. And he did the same thing with coloured flowers on the pea plants and whether the peas were wrinkly or round and things like that. So they might talk about tall and short or wrinkly round peas or the colours of the flowers. Now, he's the father of genetics, really, because he started understanding that the fact that the first generation were all tall, but there's some small came out in the second generation, they must be having some sort of units that they are being passed down from generation to generation. So he called these hereditary units. He did not know about genes or DNA at that point. So he just called them hereditary units, and he said that they can be dominant or recessive. So he was very much half of the way there in terms of genetics. He just didn't know about the genes and the alleles that were causing this recessive and dominant traits to be passed down. Other evolutionary scientists that you need to be aware of. So he was a geneticist, and then evolutionary scientists. We've got Darwin. He came up with a theory of evolution by natural selection that we talked about in detail earlier. And he went around the world studying different animals and plants and published his theory about evolution, which was quite controversial at the time because there was very little evidence. Okay, he had some fossils and things like that, but the main evidence was quite difficult for him to grasp, to prove to other people. And he published a book called On the Origin of the Species in 1859. At the same time, Wallace was also working on his theory of evolution independently. So they were studying at the same time, and he was traveling around the world, but he also came up with the same theory as Darwin, and they actually published a paper on it together before Darwin published his book. And Wallace is also known for his work on speciation and understanding how evolution by natural selection leads to speciation, and warning colours on animals, so bright yellows and oranges and stripes on wasps and things like that to warn other animals that they are poisonous. Lamarck, he was a third evolution scientist that you need to know about, and he said that changes that occur in an organism during its lifetime can be inherited. So, for an example, if I carry heavy books every day up and down the stairs, then I'm going to get strong muscles in my arms. Now, Lamarck says that because I do that every day and I'm changing my muscles, I will pass that on to my children. Okay, we now know that that's not how it happens, and that Darwin's theory of evolution by natural selection has far more evidence behind it. But Lamarck thought that if you change during your lifetime, you would then pass that down. Food security, really topical conversation at the moment. Food security by definition is having enough food to feed a population. So fishing, for example, is one way that we can help control food security. Before, anybody could just go out into the sea, catch whatever they want with whatever nets they wanted, and that was fine. But now the population is increasing rapidly. The governments have put quotas on, so it tells you exactly how many fish of each species you can catch and bring in. And they control the net size. So if you're catching smaller fish, they're not going to be very good for the food market. So you'd want a mesh size whereby your small fish, your babies, could fit through, and they will be ready to catch another year rather than just being caught and killed for no reason. And then your larger fish would be able to be caught. So controlling the mesh size is important. farming, whatever situation they're talking about, whether they're talking about chickens or pigs or any type of animal, you need to put down about the same ideas. You need to put down about restricting the movement of animals, so perhaps housing them in a small shed rather than letting them roam free naturally, and temperature controlling that room so they don't have to be wasting energy by shivering or vasodilation, vasoconstriction, whatever it is, to control their own body temperature. They can save energy that way, and therefore there'll be more energy to pass on up the food chain. And both of these reduce energy transfer to the environment. Okay, so talk about restricting their movement by putting them in smaller pens, using temperature controlled rooms, and that both of these reduce the energy transfer to the environment. The other thing that farmers are doing are feeding the animals a high protein diet to increase their muscle growth, increase their meat yield, so that we can help have enough food to feed the growing human population. Another thing that we could do is create mycoprotein. So this is an example of one of the ways we can use biotechnology, which is technology of living things. So mycoprotein is used to make meat substitutes, and it's made from a fungus called fusarium, and this is grown on glucose syrup in aerobic conditions. So the fusarium fungus then produces a protein called mycoprotein, and what they found that it's really good substitute for meat. It's high in protein, so if people don't like to eat meat, they can eat this mycoprotein, which is used in products such as corn. Another thing we can do is to try and increase our crop yield. So we talked about this before. We can genetically modify the crops that we use to try and increase the yield to maximize food security. So it could be perhaps a 6 marker on how we're going to meet the demands of the growing population in terms of food security. Trophic levels. Only a few slides left, you'll be pleased to know. Just do about trophic levels, and then we'll do about a little bit about the eye. So in a food chain, you might have these animals, for example, animals or plants, grass, snail, blackbird, and a hawk. If you're asked to draw a pyramid of biomass, it's always going to look like a pyramid with the largest base at the bottom and then going smaller, 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 because you're only looking at the mass of stuff. So when you're asked to draw one, you might be given some squared paper like this, and you just need to draw it to scale. So if I need 800 grams per meter squared of grass, if I've got 8 squares, 1, 2, 3, 4, 5, 6, 7, 8, I will use 1 square to equal 100 grams per meter squared. That's the scale that I'm going to use. So there's often a line down the middle. If not, you can draw one with a ruler. And I need to show my 800. I'm going to do 4 squares either side. So draw it across like this, 4 squares either side, and label that as your grass. Okay, that's that one done. Then the next one up is 200 grams per meter squared of snail. So just one either side like that. And that is my snail. And the next one, 50. So I just need 25 on this side, 25 on this side to make my 50. And if my label doesn't fit, I'll put that on the outside. Blackbird. And my sparrowhawk is just going to be 10. So I'll just show that by just a single line up there. And that will be my sparrowhawk. So that's how you draw a pyramid of biomass. And in terms of trophic levels, this is trophic level 1, then 2, then 3, and then 4. So the trophic levels are going up. A little bit on sexual versus asexual reproduction. I touched on it earlier, and you might need to talk about it in terms of why organisms will choose to do sexual or asexual reproduction at different times. So some things like fungi and things like that can switch between doing sexual and asexual reproduction depending on the conditions of the atmosphere. So sexual reproduction is a good choice for an organism. Think about it in terms of the population rather than individual. It's a good choice when the environmental conditions are changing. Because if you've got changing conditions, variation in the offspring increases the chance of survival. So if you're producing sexual reproduction, producing offspring that are all different, you've got variation in your population. So think about natural selection. By chance, some of them are going to be better adapted to the environment and they will survive and reproduce, keeping the population going. Asexual reproduction, however, is used when the conditions are favourable and pretty constant. So if all the conditions are good for the population, then lots of individuals can be produced quickly by just cloning themselves, and this uses less energy as well. So look at the situation that you've got in front of you. If they're changing conditions, they're going to want to use sexual. If the conditions are favourable, then they're going to want to use asexual. Last bits then, I promise you. Couple of minutes on the eye. I'm not going to go through the main bits. You can have a look at those yourself if you want to pause it or come back a little bit later on. But you need to know about what the eye, each bit of the eye is called and what it does as well. Accommodation, we're going to go through these in a little bit of detail. Accommodation is when your eye changes shape, when your lens changes shape to look at either objects that are close to you or far away. So let's look at near objects first of all. Objects that are close to you, your ciliary muscles contract. Okay, so these are muscles that are attached to your suspensory ligaments just here. They contract and your suspensory ligaments loosen or slacken, and that will cause your lens to become fat, and it will be able to refract light more. So it'll be able to bend the light and focus the image on your retina. Okay, so that's if you're looking at objects close up. Ciliary muscles contract, your suspensory ligaments loosen, and your lens becomes fat, and the light can be refracted more. If you're looking at far objects, you don't want to bend the light as much because the light's very far away. So the ciliary muscles relax. The opposite happens basically. The ciliary muscles relax, the suspensory ligaments pull tight, and the lens becomes thin so that the light's refracted less. So this lens will become a lot thinner, so the light rays will meet properly, forming an image on the back of the retina. So some people that find it difficult to change the shape of their lens might need to wear glasses. And you might be short-sighted, which means that you can see things that are close up, but struggle to see things that are further away. And this is called myopia. Now, with short-sighted, you need to treat it with a concave glass because you're bending the light too much. You can see the things close to you, so you're quite happy with making a fat lens and bending the light. So to help you see things that are further away, you need to use a concave lens on your glasses. The opposite is true for long-sightedness. You can see things far away, but you have trouble making your lens a nice fat round shape to look at close objects. So if you treat this with a convex lens, that will help refract the light and help you focus the image and take it from back here to actually refracting it more and forming on the retina. Final couple of slides. In relation to the reflex and responses that we talked about before, in terms of nervous responses and automatic responses, when your eyes are suddenly put in front of bright lights, there is a reflex response to protect your eyesight. In bright light, your radial muscles relax. So this is the reflex bit, the 3 R's. If you're reflex, because you've got a bright light in front of you, the radial muscles relax. R. Reflex, the radial muscles relax in bright light. The circular muscles contract. Now these aren't shown on here. These are if you look at the light head on like that. So you've got your radial muscles and your circular muscles. So they contract and it controls the size of the pupil. So in bright light, you want that pupil to go really small so that not much light gets in your eye. In dim light, the opposite happens. Radial muscles contract, so these ones contract, and the circular muscles relax, and that opens up and dilates your pupil, so as much light as possible can go in, and you can see in dim light.